you're going to have to split open the DNA strand, but remember we've got supercoiling issues. So how can we fix that supercoiling issue?
Oh, yeah, I forgot.
You forgot about the supercoiling?
Well, I remember you talking about it.
Yeah, so as you start to pull it apart, we're going to get supercoiling on the front end and on the back end of your replication sites. So how can we fix that stress? Was there, say, an enzyme that dealt with supercoiling? What was that enzyme?
Gyrase?
Yeah, your DNA gyrase. I actually kind of forgot it too, so that's why I was hoping you could help me out. So the DNA gyrase will help alleviate that supercoiling stress, right? Because yeah, as you pull that open, you are going to build a supercoil within that, and that's going to ultimately then prevent your replication from continuing forward because it's now so coiled that it can't unwind anymore. So you do need that process to relieve that supercoiling. All right. What's the next part you're going to need?
Your single-stranded binding protein.
No, not even that. So the gyrase relieves the supercoiling, which is good, but then we still haven't split open the double helix. So we also have to split open the double helix. How do we split that? That was our helicase. The helicase will then unwind it, and now we do have these single strands. And now we've got an issue because within the nucleus, or even in, what is it in prokaryotes? Not nucleolus. Is it nucleolus? Oh, okay. I guess I had that right in my head. We've got a high concentration of nucleases, meaning they will go through and they will cut apart strands of DNA, okay, ultimately single-stranded things. Okay, so as soon as we've unwound the DNA, now those can potentially get cut. And we don't want them to get cut because that's where all our genetic information is stored. So that's where we can bring in the SSBs, which was the single-stranded binding proteins, which can lock onto the DNA and now kind of prevent the nucleases from attacking it. So that can help protect it, but then we still have to go through and replicate. So how are we going to start that replication process? We're going to have to be aware of how our strands are going to be synthesized. So we're always going from 5' to 3'. So as we unzip, we're going to start to expose a new 5' end, and we can start to replicate that. Okay? So DNA always copies in that transition. So we're going off of our template 3' to 5' to generate our 5' to 3'. That's going to be our leading strand. So as soon as a new nucleotide is exposed off of that strand, we can put on a fresh nucleotide from the solvent, and our polymerase can help with that process. All right, sliding it on there. What then becomes the natural issue? What about the other strand? The other strand is now exposed, and because polymerase can only run in the 5' to 3' direction, as soon as we pop open a new section, that's not giving us the ability to actually slide or synthesize in that 5' to 3' direction. So what ends up happening is that lagging strand ends up having to get folded back and go through a slightly different processing. Right? In that processing, we're going to use what they're referring to as your Okazaki fragments, and we'll start to fill in that synthesis off of those strands with those larger sections. But in the process of doing so, you'll notice that they leave a bunch of little marks. What are those little marks? What's happening in those cases?
There are actually little breaks.
They're breaks in the
template strand.
Not in the template. The template's fine.
I mean, it like comes and goes off. It detaches and does like sequence and then it comes off.
True, but what does that break mean? Why don't they show it a nice continuous area? It's a gap. What we're looking at is a gap in the phosphodiester backbone. The polymerase can't seal that. So all it does is slide in the nucleotides that match into that fit, and it can kind of fit those as they chunk out. But between our two sections, between one fragment and the next, we have an issue. The polymerase can't seal that gap. Okay? Because remember, it can only go 5' to 3', so it actually doesn't see those gaps. It ends up stopping and says, well, I can't deal with that, and it spits it off, and it can start a new loop. All right, so how can we go back and seal those fragments? That's where we use DNA ligase. The ligase pops on and goes through and fills in those gaps. Okay? So that's what they're trying to show with this loop. So when we look at just a standard format, it looks all nice and neat. To go through and actually synthesize the lagging strand, it'll actually pump it back and loop it around, which then means when it passes through your polymerase, it does set up in that 5' to 3' direction, and it allows it to polymerize that a little bit cleaner. Okay? So it's kind of a convoluted process when going through and setting that up. With your polymerase, we have all sorts of different types of, or was it 5 different types of polymerase that we found, at least in prokaryotes. For it to function, we need all of the nucleotides, because the nucleotides have to be incorporated in. So if we don't have those nucleotides, it's going to cease functioning. We'll need magnesium ion. Why magnesium ion? Magnesium ion is probably contributing to our tertiary structure of that polymerase enzyme. Without that magnesium ion, it's probably not folding up properly, which means it can't interact with the DNA chain properly. So that goes all the way back to our tertiary structure chapter in 3 or 4. Okay. And then it also needs an RNA primer. So our DNA polymerase can't just start throwing on nucleotides. It needs something to actually have that nucleotide attach to. That attachment point or initiation point is your RNA primer. So, and a strand of RNA will match with your DNA, fit up there, so it can now say, oh, I can attach to that point. I now have my complementary nucleophile electrophile system, and it can start to build outwards. Okay. What's going to be an issue with an RNA primer? It's RNA. So what's going to be one of our problems? After we go through and synthesize, we're going to have to double back, kick out the RNA primer, and put in real DNA. Put in DNA. So your different types, if you have polymerase 1, which just happens to be the first one found, which is why it came up with 1, it's actually a repair enzyme. So what it's going to go through and do is repair and patch any issues within DNA. Its primary function is repairing and patching the RNA sections. So it'll pop those out and replace those with DNA. Polymerase 3 polymerizes the new strand. Okay, so it's the one involved in actually making that new strand copy outwards. Polymerase 2, 4, and 5 are all going to be proofreading enzymes. Okay, so proofreading and repair. So once we've synthesized it, we have to make sure we didn't make any mistakes. So those polymerases will double back through and say, okay, whoop, there's a mistake. And then we have to go through and fix that mistake. All right. So if we put it all together, we can get a nicer image coming out of this. And I've got a couple movie links, because there were some requests for movies. So we've got some YouTube movies that look actually somewhat interesting that might be helpful. What's that? Turn on the lights?
No, off the lights.
Yeah, we can turn off the lights. So we can see our DNA gyrase, which is going to be doing what? What is the DNA gyrase going to do for us?
It's going to prevent that.
Make it tighter.
It's going to fix our supercoiling issue stemming from our replication. We have the helicase, which is actually going to allow it to split the double helix. We will have some of those single binding, I keep calling single binding, let's just say SSBs, just temporarily stabilize those to give us that space so that we can then actually put on our polymerase. There's different models for it, and I'm pretty sure our polymerase is actually a trimer. We have 3 different sections of polymerase within it. So this is active research, so there are people that are adjusting and saying, okay, no, we need this, we need this. We don't quite have that figured out. Okay? And then your lagging strand has to do that loop back. That loop then goes back through a different polymerase polymer, which then allows for the extension of that chain. Okay? So we've got all those kind of pretty pieces shoveled onto that. Is it correct the first time? Well, no. We're going to have errors in that replication. And depending on the type of error, we get different error percentages. So my first pass through, I saw this 10 to the 9th or 10 to the 10th base pairs. So every 10 to the 9th or 10 to the 10th base pairs, we get a mismatch in the DNA. Okay, that's pretty insanely accurate if you ask me. So it's kind of an impressive process to be able to go through and do. That means that everybody in this classroom, when attempting to synthesize the DNA strand in the back, cannot make a single mistake. Right?
Yeah.
And realize that, remember, in this process, we've got solvent actions, we've got nucleotides that are potentially hydrogen bonding and getting in the way. It has to kick those out and allow for the processing to continue forwards. Okay, so there's a lot of different things that can ruin it. So to get such a small error percentage is pretty impressive. So we will definitely have to go through and proofread our cells or proofread our DNA to check for those mistakes. So even that small of a mistake percentage does generate a very toxic or potentially toxic effect on the cell. So just a single nucleotide difference, I'll walk further away from you so you can open that. Just a single nucleotide difference can end up resulting in cell death. So we need to come up with some way to fix that. Even though we're looking at a massive amount of base pairs, it is really important to make sure that we maintain that fidelity. And then we can also look at hydrogen bonding errors. So if we get a mismatch in our hydrogen bonding, it's a slightly different error than our mismatch coming from the polymerization, and that happens a little bit more frequently. So we still need to be able to fix all of those errors. Even with that one happening more frequently, 10 to the 4th, 10 to the 5th is still an incredible percentage, low percentage for error. All right. So there's a variety of proofreading repair mechanisms. You'll get asked some questions on sapling, and I've added some slides into this to make sure you can pick up those questions. So a variety of different setups, and there's a lot of overlap between those. For instance, the cut and patch mechanism is very, very similar to the NIC translation, okay, as far as the overall comparisons and when they pop up. So your cut and patch, polymerase 1, will go through and cut out the primer, so our RNA primer that we didn't want there, and it'll patch back over it with the proper base pairs. Okay. The NIC translation happens, instead of cutting out the RNA primer, we're looking at a mismatch or a DNA mistake. So what it'll go through and do is it'll cut out that sequence of DNA that has the mistake and then backfill with the proper base pairs or attempt to replicate again to fill that base pair hole. So it's very, very similar. It's just looking at slightly different processes. Your cut and patch is, I think, a little bit more pushed towards your RNA translation than your DNA, or not RNA translation, RNA primer versus your DNA mismatches. Okay? Mismatch repair, in that case, if we have a mismatch between our bases, our base pairing, we're going to get a different process going through and finding that. That's actually going to involve different enzymes that are going to identify that mismatch, then remove and re-replicate that section. So it's not tied to your polymerase 1. And then your last repair method or type of repair is base excision repair. And in that case, what we're looking at is DNA glycosylase, glycosylase, if I look at it, makes it easier to say, which will remove the damaged base. And then your sugar and phosphate at a slightly later stage, okay, and then it can backfill with your polymerase 1. Okay, so we've got all those potential types. If we go through and look at our cut and patch, it can identify, or your polymerase 1 will identify that RNA primer, pop it out, and then again backfill with your DNA. What's happening with the NIC translation is that where we'll have a NIC, so this is what we were talking about in that lagging strand, okay, we've got a section there where we can't seal that up because our polymerase doesn't fit it nicely. So what the DNA can go, or our polymerase can go through and do is identify that NIC, pull it, and then go through and backfill and seal, and what ends up happening is our NIC ends up translating further down the DNA strand. So that's where they're getting the NIC translation. Because once it pulls it out, it ends up translating that NIC to a different spot where it still can't fix it. Okay, so a new NIC translation would have to come in, and we'd have to propagate and fix that error all the way up. Okay. We can get our mismatch repair, and this is where we're looking at not polymerase 1. We're looking at an enzyme coming in and fixing this. So if we have a mismatch alignment, so in this case we've got T and G, we're going to have a distortion in the the double helix backbone. That distortion can then be picked up by enzymes. Those enzymes have been labeled mut H through a bunch of other different letters, which is looking at potentially for mutation would be my guess. And what those enzymes are going to do is find that site and then go through and remove that section of DNA, okay, the bad one. Well, how do you know it's a bad one?
Read it.
How do you read it? You've got 2 strands of DNA. Which one's bad?
The non-parent one.
Okay.
The red one.
There we go. We can get a little bit of color into it, but how do we know, or how would the cell know that the red one is good versus the blue one being bad?
The blue one is methylated?
If we go back through, if you take a look all the way through at this, we see a methylation site. What's happening with that methylation site?
It flags it.
That is marking that strand as the
New one.
original strand. Okay? So that when your mutation occurs, it can say, oh, here's a mutation, but now which end of the strand do I need to fix? If it doesn't know which strand is bad, it can't fix that. So that's where another sequence of enzymes will go find the nearest methylated site, which marks our DNA strand as the original. It will link those 2 enzymes together, and now it knows, oh, okay, because we're connected in this fashion, that strand is the good one. So what it will then do is cleave off the other strand, the entire other strand, okay, between those 2 sites. It can then backfill and put in the proper nucleotides, thank you. What is it filling with? Okay, and that's where our polymerase can come back in and help out. We'll still need the ligase because we still have to close that gap, that phosphodiester backbone. But here is so.
So it removes only from the methyl group that have in this state?
Yes. Okay, kind of a neat process. A lot of this, I will admit, is entirely fascinating, but to me it's just a bunch of facts. What I think is even more interesting is how experimentally they're able to figure all of this out, okay, which I don't have the time to figure out at this point. So we'll just have to talk about those facts, and if you take the class next year, I'll give you more information. The last one we want to kind of cover for our repair, primarily for the sapling homework, in case you haven't gone through and done that yet, is your base versus nucleotide excision repair, because they're slightly different processes. What's happening in base excision?
The base has been damaged by oxidation.
What part of the nucleotide is being removed in base excision?
Just the base.
Just the base. Okay, so your adenine, your thymine, your cytosine, your guanine. Okay, it's not touching the deoxyribose sugar or the phosphate backbone. Those are left in place. So in base excision, it'll identify the mistake, it'll remove the base, but now we're missing a big section or a section within our DNA, and then we can go back through, remove the ribose and our phosphate with a new enzyme. Once we've got that whole, we can then go back through and fill in with our proper nucleotide, and that goes back to our polymerase 1. Okay, so we're looking at a sequence of steps to go through and fix that mistake. So base excision, remove the base, then a new enzyme comes in, removes the sugar and the phosphate, and then polymerase 1 goes, oh, there's a hole, and it can then go through and fill it. Okay? In the nucleotide excision, it's a slightly different process. What's the difference between a base and a nucleotide? That's a question you should be able to answer, because we went through that nice and slow at the beginning of this. The base is just your pyrimidine or your purine. Your nucleotide has the sugar attached to it, and then we could even go and take it a step further. We have the phosphate attached to our nucleotide. Okay? So in the nucleotide excision, what ends up happening is we recognize the mistake, and then this, was it exonuclease, the ABC exonuclease, comes through and removes a chunk of bad DNA, including the mismatch or the improper nucleotide, and then polymerase 1 goes through and fills, we still have to fill that, or sorry, backbone. We still have to seal the backbone, and that's where our DNA ligase will come back through after the polymerase puts in the proper nucleotides to fill it. Okay? So in your base excision, we're pulling out just a single nucleotide effectively. Okay? In the nucleotide, we're pulling out a pretty large chunk of DNA. Okay. So if we look at the picture that I've got supplied, if we were going to look at base excision, we're really just removing that section. Then we can remove, come on, it is drawing, right? Yeah. Then we can remove our ribose and our sugar, and then we can go back through. Our exonuclease sees that hole and wants to make it a little bit bigger, so the exonuclease comes through, chops out a couple more nucleotides so that our polymerase 1 can recognize the hole, fill it with the proper nucleotides, and we can seal that up. In the nucleotide excision, we recognize the mistake. We say there's a mistake, let's just kill everything. And now let's go through and fix it. Okay? So those are your primary differences. You do have to watch out for the different sugars, or sorry, the different enzymes that are involved in each of those processes. Yes?
What's going to be the main thing, well, other than, is it the problem in the base or the problem in the nucleotide that'll determine which one will happen?
Yes. And it's going to depend on the process, probably on what it recognizes. Because when we're looking at the nucleotide, we're really not looking at very many options for it being wrong. The issue for it being wrong would either be the wrong sugar or it's the wrong base. So it's probably just what it recognizes first. Which of these enzymes says, oh, that's a problem, and fixes it first. That's my guess. Unfortunately, that's all I got. We do get a little bit of genetic freedom within this. So if we just copied our DNA straight across, if something drastic were to happen in our environment, we don't really have a way to adapt and change. Right? So there is some ability to allow for natural mutations to occur. The issue with mutations also leads to cell death, okay, potentially. So, but if there's a massive catastrophe where we have to go through and adapt, we need some way to affect a change on the DNA to allow for that mutation to actually propagate through and potentially allow for survival of the cell. Okay? That process comes in when we're looking at this kind of crossover. It's not crossover. What's it called? There it is. Genetic recombination. Okay, close enough. Crossover recombination. So what ends up happening is we will go through and swap strands of DNA, cut things and flip them around, and that allows for the potential for different mutations to occur in different transfer genes. Okay? So we do have that possibility happening with our DNA as well. So you can refer to homologous or non-homologous. If you're going to take genetics, they're probably going to talk about that a whole heck of a lot more. Okay? Beyond that it exists, that's all we're really going to discuss. Okay? Eukaryotic replication. What's our primary difference between prokaryotic and eukaryotic? What's that? We've got a nucleus. What was that?
Circular versus linear?
As far as the DNA, we're going circular for our prokaryotic, and eukaryotic is stranded or linear. That's why you said linear. The other big issue is that it's a lot more complex. Because we have a much larger increase in complexity, we also know a whole heck of a lot less about it. Okay? So they've gone through and looked at those. Yeast is going to be the most, the primary one studied. Why look at yeast and not say humans? Okay, we're again trying to look at complexity. Your yeast is a lot more simple than your human DNA replication, and it's also very fast replicating. So we can propagate through, and we can also look at cost. Okay, so it gets divided up into different phases, and we have a whole lot more polymerases involved in this process. Okay? I don't, again, particularly care much more beyond that. We've got the general idea on what has to happen. We still have to split it open. We still have to polymerize it. We still have to go through all those individual steps. So those same complications that we run into with the prokaryotic DNA, we run into with the eukaryotic DNA as well. And we can look at the different phases on when those happens. And that's when we're looking at cell growth, the whole G1S, G2M phases, which you probably know a lot more about from your biology classes. Okay? When we move up to eukaryotic DNA, the one thing, just because I like pretty pictures, and they happen to have some nice pretty pictures, polymerase 3, which is our primary DNA replicator in eukaryotic DNA, is replaced by a new enzyme called PCNA, which is the equivalent polymerizer. Okay, so they've got kind of a nice three-dimensional X-ray crystallograph of this, so we could look at the modeling on the left where we can see all our helices and we can see how those things fold. So you've got a lot of alpha helices. I think we even have some beta sheets popping up in here that they didn't label quite as nicely. And then we could also look at what the X-ray crystallograph would image for us. Okay, so it just looks kind of neat. The DNA is going to sit right through the middle of that, and that's going to help facilitate the polymerization process. I'm going to split that open. Okay. Yay, differences in our DNA and prokaryotic systems. Okay. We already talked about some of those. We've got some more polymerases. They went through and listed a bunch of those different roles. We have some primary differences between our polymerases. So when we jump all the way to our polymerases, in our prokaryotic, they can act as exonucleases. In our polymerases, they can't. Or in our prokaryotes, they can't. We can look at different sizes of the Okazaki fragments. And we can also look at where we origin our replication. Where our origin of replication starts. In prokaryotes, we have one origin. In the eukaryotes, we have multiple different sections, or multiple different origins within the strand of DNA. Okay? Another thing that we didn't talk about is with our eukaryotes, we've got histones. Those histones are complex to the DNA. That's going to make it an added wrinkle on how we're going to go through and process that DNA. Okay? And again, remember when we talked about the histones, we don't know exactly their role. They could be facilitating or hindering that process. Okay? We already mentioned this. Our biggest difference, I think, between our prokaryotes and eukaryotes, the one that's going to be most important for us to lead into the next section, is that circular versus linear strands of our DNA. Okay? Which on its own may not seem like a bad or a big issue, but when we look at our DNA strands, we've got nucleases that are constantly floating through the environment. With a circular strand, we can supercoil that up, so the whole DNA sequence is all folded up on itself, and there's no real easy spot to bite, okay, for our nucleases. What happens with a single strand? Well, that single strand has a beginning and an end, and both of those spots we can't hide quite as well as we could in prokaryotes. That means what could happen with our nucleases? Our nucleases could come through and potentially cut off those top and bottom strands of our DNA. Okay? On its own, that may not sound like a bad thing until we consider what our DNA is coding for. It's effectively coding for our life. If we go through and start cutting off sections of DNA, there goes our life. So we need to have some way to fix that, okay? Or to either prevent that or some other process. So the way we do it in eukaryotes is we introduce this telomeres. Okay? So our DNA was intelligent enough to slide in these extra strands of DNA that are just fluff. They're there in case our nucleases come through and start cleaving things off. Okay? This can then stabilize our DNA and allow our life to continue on. Not indefinitely, but potentially extend longevity of the cell, okay, or that DNA genetic code. What happens if that telomere starts to get cut off? We could now start to die or increase the likelihood of mutations. Okay, remember, our mutations have to do something about a change in the DNA sequence. So if we can remove that telomere, we might actually potentially increase mutations. Why might we want to increase mutations? Survival. When is survival going to become important?
Nutrition is low.
When nutrition is low, what else could potentially cause it? Big temperature changes, pretty much anything that is stressful. Okay, so if we shock our cells and get them to freak out enough, what's going to end up happening is our nucleases are going to increase their activity, and we're going to start losing those telomeres, and we could potentially start to increase our mutations to hopefully allow for the longevity of our genetic code, that it persists or somehow changes so that we can actually continue to propagate that forward. Okay? So there's a lot of active research when looking at our telomeres, and when they first talked about it, they really look at it as kind of a fountain of youth. The longer our telomeres are, the more likely that genetic information stays constant, and we could potentially live longer. They also found that the older you get, the shorter your telomere length goes. Okay, so they were saying, well, if we just make sure that our telomeres are always super long, then we'll never die. Okay? And there's some truth to that, but it's not perfected yet. So how do we get those telomeres to be longer? And that's what they're trying to show with these big, long, little red dinguses on the end. Okay, those are our telomeres. Dingus? Yes. Feel free to look it up. Just not during the class. So we need to be able to extend that chain. To do that, we need an enzyme to go through and process that. That's our telomerase. Okay? So within the telomerase molecule, it's going to contain a strand of RNA. That strand of RNA is going to code for the telomeres. Okay? And we really don't want any genetic information in that. We just want a bunch of nonsense so that if the nuclease comes along, it doesn't completely destroy our genetic information. So what our telomerase will hold on to is the information that matches our telomeres. Okay, so it's complementary. Ooh, I'm not sure if that's the right sequence. I think that's the RNA sequence. Your textbook does list both of them. It lists what the RNA sequence is and then the DNA code that comes with it as well. Okay, and I may have copied the wrong one because I don't remember. What's that?
You got a U in there.
Oh yeah, you're right. So we did get it. When it copies into DNA, what would we expect to see in it? You should expect to see that TNA or T show up in that. All right? So your telomerase holds that information so it can go through and put that on. How many I keep calling them amino acids, nucleotides, do we need coming off of our telomeres? What is the proper length? All right, we're looking at thousands. So this, and not just thousands of nucleotides, we're looking at this repeating unit happening thousands of times. All right, so we got a lot of extra space on there so that if our nucleases come through and start cleaving it, not a big deal. It's not going to eat into our genetic code. Okay? Questions about that? Or any interesting, cool things about telomeres? Yeah.
It's a question. I was wondering if like anything has been theorized as far as like, okay, so what if we did have like the linear sort of DNA like bacteria, or would that even not even work?
Or circular? I don't know if they theorized on that. I'm not sure how you would set that up, or why necessarily circular only goes with single celled versus the other way around. Yeah.
So with telomeres, they have a chemical expansion of telomeres by injecting human growth hormones. So
Oh, you were talking about all those studies, yeah.
yeah, so that's how you would do it. So they'll I think it's like $1,000 per week to have human growth hormone to do that. So a lot of people.
Oh, this isn't just a study. This is some crazy people injecting
people actually
random.
do it. So they'll see a 60-year-old less wrinkles because they think it's less harmful than using plastic surgery. So they use human growth hormone. It's $1,000 per week for the treatment of that.
So.
It drives for every week because the telomeres are doing such a rapid rate because you're having longer ones, so now that's increasing. So that means you're having piezoelasticity to your skin. This is due to cell reproduction. So if you put on more telomeres, you have more nucleases that will actually cleave. That means you're increasing your cell production. So that's how they do it. They put human growth hormone.
Love the idea of increasing your cell production. There's another way you can do that. Cancer.
Cancer, exactly.
People will do anything to get through aging. So we've got some videos. Which one do I want to look at? I think I want to look at this first one. which is probably going to play only on my computer. No, cool, put up here. Yay, ads. Yeah, let's go ahead and skip that. Oh no. Friggin' Pearson. Oh, and they threw in a bunch of other junk in here too. So we can zoom in on the DNA. Oh yeah, we wanted the lights off, right? Wrong way. Still wrong way. There it is. So what they're trying to show here is that we've got our origin, so we're starting to split that DNA open, and we can start to pick out, so I can pause this. So we've got our DNA that's now getting threaded through and getting split apart, and we've got this trimer. You'll notice all three of these pieces are connected to each other, and we've got one strand that's replicating immediately outwards. Okay? So as soon as it unzips, we can slide in nucleotides and we don't have any issues. What happens when we shift to our other strand? You'll notice that it stays single-stranded all the way out until it loops back to another one. Once we put in a new fragment, it can then start replicating outwards, okay, in that proper direction. Okay? And it's going to continue to replicate back this direction until it encounters the previous fragment. Is it going really slow on your computer? No, just mine. Okay. So it's going to continue to propagate outwards. Meanwhile, we have this big long strand that could potentially be cleaved. Okay. As soon as it goes through and hits the previous RNA primer, it now says, well, I don't know what to do anymore. So that strand can then loop back down and around, link back. Fine, show the other one. There we go. And it's going to have something help facilitate getting that in there, and it can then start cleaving that, or not cleaving, start sliding your nucleotides back into that process. And you'll again notice that we're still not replicating our other strand. Okay? And it's because we have to synthesize from our 5' to 3' direction.
Were all those exposed? Right?
All of those are officially exposed. What they aren't showing within that process would be the SSBs, probably linking to that and preventing nuclease activity. Okay, so now they're getting in there, showing it all fancy, going through and trying to connect those individual amino acids, nucleotides, into our sequence, and it's polymerizing that process all the way through. So that's going to be our polymerase 3. Okay, within the lagging strand, again, we have something to facilitate, and it's going to loop that back around. It's going to put on your RNA primer. So what was that enzyme likely that just left the screen? Our primase, okay? That's there to slide in our RNA primer, which then allows for our polymerase to actually function. Because remember, it has to have something to link that DNA to. If it doesn't have that bond, it can't go through and process. And again, it butts up against the new process, or the next one, and it now can't seal that. So we have what enzyme? We've got a ligase, which is not officially happening here. Because what's happening? We're getting rid of the primer. So what's our yellow enzyme likely? It's not ligase. Ligase isn't a repair enzyme. It's our polymerase 1. We've got our gap. What's the green enzyme now? That's going to fill our nick, and we now have our ligase that came in and completed that process. All right? So kind of an interesting process. We talked about some of those enzymes, and some of them we didn't. As it continues to propagate outwards, we can then replicate all of our DNA. Kind of a neat process. And sort of cool video too, right? Okay. Let's see, can I get back to that one? Let's try that next video, I think. I don't know how well this one's going to show up. It was kind of cruddy on my computer. On my computer. Let's see if that helps. I may have muted.
In this animation, we'll see the remarkable way our DNA is tightly packed up so that 6 feet of this long molecule fits into the microscopic nucleus of every cell.
I don't care.
That's good.
I don't care.
They form around the time cells divide when the 2 copies of the cell's DNA
There we
need to
go.
be separated.
Starting to get in the right process. I think this is where they actually start the replication process. Yep.
Using computer animation based on molecular research.
See, theirs isn't quite as nice.
How DNA is actually copied in
Actually,
cells.
I think looks pretty nice actually.
You are looking at an assembly line of amazing
So you can see that
machines that are
chain getting pushed out and it can't really replicate it.
The DNA to be copied enters the production line from bottom left.
Where's the leading strand?
Molecular machine is called.
They're both to the right. Well, never mind, it's in the right here. Sorry, I was thinking the previous image. Where's our leading strand? On the bottom. You can see that loop again propagating outwards until it butts up against the previous RNA primer, and then it gets kicked off and we have to loop that strand back through.
Because it must be copied backwards. It is drawn out repeatedly in loops, then copied one section at a time. The end result is 2 new DNA molecules.
Beautiful. Okay. We'll save that for probably Wednesday. Wednesday? Tuesday? Thursday. Whenever we next meet. Okay, so now what I think would be kind of fun to give it a shot and see how well this works is what we've got in back. Our game. Our game-ish. I said I was going to set up 2 different copies, and this took all day Sunday, so I wasn't about to make another copy. So what we've got set up is our DNA strand to begin with, sitting right in the middle. And we're going to have some issues with this replication process, because you'll notice that I can't really have one of our primers running the other direction very easily, because we get a hanging issue. So I'd have to generate a stick to hold up the bottom of the other one. So we just kind of have to work with what we got. So the idea behind this is that we need to go through and replicate. So one of the first things that we could do is show that helix. And it actually holds, depending on which partners I've got. Hope I'm not silver-coating. We can actually show a little bit of a helix on that by twisting it. So the first thing that our DNA would have to do, untwist. Now that we're in a somewhat stable untwist, now our helicase could go through and potentially split this open and start the replication process. Right? So we can attempt to give someone a chance to do this not blindfolded and see what happens. But before we go through and do that, it might be nice to discuss what's happening within our individual nucleotides. Right? So with our nucleotides, I did try to set this up as cleanly as I could. We've got our nucleotide, our base, which is going to have our hydrogen bonding interactions. Right? Yeah, I could go through and do all the numbering and properly line them up. Styrofoam doesn't cut well, so deal with it. Okay. Our nucleotides are differently paired, though. I did set that up. So, for instance, back here, I can't pair these 2. They aren't going to set up a proper connection.
Out the door.
I don't know what's going on. I'm not sure I'm going to find it.
Wow.
Well, if anybody tries to jump through the giant window, I think they're going to get hurt.
Right? That would actually be kind of